Welcome back to Expert Insights by DCN Diagnostics. I'm Mitzi Rettinger. Today I'm talking with Rob Rich, Director of Sales for the Americas at Biodot. If you work on lateral flow, you know the jump from a good prototype to a stable line is where a lot of programs win or lose. Today we're going to focus on real problems that Biodot helps teams solve, what users actually value when they're on the equipment, and what Biodot plans to highlight at ALFC this October. We will close with Rob's advice for people selling into this technical market. Welcome, Rob. It's nice to have you join me today.
Hey, thanks for having me. It's my pleasure. Been looking forward to it.
Great. You know, at DCN Diagnostics, we see a range of platforms, processes, practices in the field. Today, we're going to focus on Biodot's view of good manufacturing processes and how teams make smart early decisions. Rob, to get us started, can you give us a quick scope of your role, including which customer profiles and workflows you support day to day in the Americas?
Yeah, so as you mentioned, my title is Director of Sales for the Americas. I've been with Biodot now for over 9 years. Most of that time, I've been exposed and working with the lateral flow industry in general. A lot of the work that I do is on the manufacturing side of lateral flow tests. So how you're striping, spraying, and manufacturing the actual lateral flow rapid diagnostic test is something I spend a lot of time thinking about and understanding the certain customers' challenges and maybe the unique applications that they might have.
So, Rob, you said you've been at Biodot for 9 years. Tell me a little bit about Biodot.
Yeah, interesting you should ask. So Biodot was founded back in 1994 by a gentleman by the name of Tom Tassone. And so we've been actually doing lateral flow test manufacturing for 32 years now. I think we probably have some 20 to 25-year-old equipment still out there functioning and working. So we're pretty proud of that. We feel like we know what most of the challenges are and how to address them.
So, Rob, I don't know. I know you know, but I'm not sure if our listeners know that DCN was actually founded. Our founder was a part of Biodot, and DCN was founded in 2006, coming out of Biodot to form our company.
Yes. And the partnership has been fantastic ever since.
Yes, it has. In one sentence, what problem does Biodot exist to solve for those diagnostic manufacturers?
Yeah, so the idea here with Biodot when it comes to the lateral flow manufacturing equipment that we manufacture is to not only support R&D level development work, but honestly, we're attempting to cover the entire ecosystem. So we want to go from R&D all the way to full scale manufacturing using the same exact dispensing technology and make life easier for the end user and customer due to the fact that it'll streamline validation and technology transfers as they progress through the different types of manufacturing processes and scalability.
That leads perfectly into my next question. So why don't you walk us through those core steps that you see on most of those LFA lines? So where do errors creep in the most often, and where do strong teams, what do they do to prevent that from happening?
Yeah, so the most important thing other than the development work that you there at DCN do yourselves is once you've developed your assay, you need to have a high quality dispensing system to lay down your test and control lines in both qualitative or quantitative manner. We do the same thing with spraying conjugates as well. And I think the biggest mistakes we might see is that people take for granted that the performance of your test and the sensitivity of your test will be driven by the quality of the dispensing that you are doing onto your nitrocellulose. And so if you're not really willing to put in the time or actually make the investment into a high quality piece of equipment, then you're going to spend a lot of time working through challenges that may otherwise have not existed.
That makes a lot of sense. We've have several clients that come to us and they say they're ready for manufacturing. And usually once we take a look at it, we see more likely they're really ready for scale up and optimization and not quite ready to make that leap. And it kind of leads into what you're saying, that upfront development time, like taking time to get the right equipment, spend time
Correct.
in the R&D portion before you make that leap. So from your point of view, when there's a program that's still in development, what really needs to be locked down before they say that they're ready for that real manufacturing run?
You have to have every piece of material selected, whether it's the thickness, the width, the stack up, the overlap of the materials on the backing card. You really have to have everything absolutely locked down, and you have to know exactly how you want to manufacture it. So if you're not ready to make a commitment to buying 100-meter rolls of material to the exact specification, that you need during the manufacturing process, and you're probably not ready to scale up. You need to spend more time getting those aspects set. And then at that point in time, you will likely be ready. And then to also add to that, it's not just locking in the materials, but I also tell a lot of my customers that if you're not going to be manufacturing 1 million plus tests annually, then you might not need to move to the high scale inline manufacturing just yet, because it is a large capital investment and you have to have a large commitment from a facility standpoint and personnel standpoint to support it. And so if you can't do that, then maybe you want to continue to leverage contract manufacturers until you are in fact ready.
So really, a lot of manufacturing can actually be done on those benchtop models based on the overall yearly.
That's right. Exactly. Yeah. We figure, at least 300 to 500,000 tests annually on a single batch manufacturing setup. So you need an XYZ striper and sprayer. You need a laminator and a guillotine cutter. And you can do a lot of damage in manufacturing quite a few parts on those systems. Not until you approach the 1 million-piece mark is when I begin to engage and have serious conversations with the customer and streamlining their manufacturing process.
Got it. I think, maybe where this line is when we're talking about manufacturing, it's not necessarily that jump from benchtop to large standalone equipment. It's more about saying, have I spent enough time in research? Do I have everything locked down in my materials? What I want to do? Now I'm going to optimize the assay. Am I optimizing it on the benchtop, or am I optimizing it, shifting over and optimizing it and scaling it up on a larger piece of equipment?
Correct.
You still have to do that optimization. You still have to do that V&V. It's just which piece of equipment am I doing it on?
That's extremely accurate, actually. This is one of the benefits of dealing with BioDot is we absolutely utilize the same dispensing technology on a benchtop system for batch manufacturing as we do in inline. So your settings transfer over from your benchtop to your inline system beautifully. The only difference usually is how you're going to be drying the materials, right? In batch, you've got like a convection oven in your lab, and when you go to inline, we have inline drying towers. So you have to do a little work there to figure out what the proper drying times and what the speed or the rate of the web material going through the drying tower should be. But other than that, you don't have to worry about the dispensing side of things. It transfers over because we don't skimp on quality in anything that we do, from benchtop all the way to the high scale manufacturing line.
That's great. So maybe if we step back a little into that more of the research, what am I going to do next phase? When teams are trying to choose between dot disposition and continuous line dispensing, how do you guide their choice so that the process fits the assay?
We look at it wholly as a manufacturing challenge. Of course, we want to have high quality dispensing so that we're delivering very high precision volumes into or onto the nitrocellulose membrane. A few challenges that I see, a lot of people do come to us about doing like a dot matrix type pattern onto a nitrocellulose where we're spotting an array or a pattern of some kind versus a simple line. It sounds excellent in theory. It's a little more challenging in reality because of the manufacturing processes that you need to think of downstream. So I tend to point that out up front because you do need to think about it if you're going to scale. And then, what's the cost, the ultimate cost of the lateral flow test, and does it warrant moving from your typical traditional lines versus spotting? Because there's going to be added cost on the capital equipment side and probably additional material costs. The reason I say that is if you think about it, when you stripe your nitrocellulose, right, and you have a 30 centimeter card or a 100-meter roll of continuous line being striped, you can set the downstream manufacturing systems that are cutting the materials and laminating the materials up to cut anywhere on that card and just make sure that the width of the strip is exactly what your call out is, whether it's 3.5 millimeters, 4 millimeters, 4.5 millimeters. So it's really straightforward. You simply move, cut, and do that at any specific distance that you so choose. But when you're laying down a pattern onto the nitrocellulose, you now create a situation where you've got to get that pattern to line up perfectly within the window of your cassette or cartridge. So you need to know where the array pattern is on the nitrocellulose. And of course, there's spacing from one array pattern to another array pattern. So then we need to add an extra dispensing step where we're maybe putting down a fiducial mark, a visual mark, right? That we can then trigger a cut downstream and make sure that we actually know where the array was printed. Because once your reagent dries onto the nitrocellulose, you can no longer witness it or see it. So those are the little nuances of considering one over the other. Naturally, a lot of people gravitate to the array printing only because of the fact that you can do maybe so much more multiplexing on a single test strip. But as I'm sure DCN is aware, and you guys being, the preeminent development house in the lateral flow industry, you got a lot of other considerations when it comes to conjugates and getting them to all play nice together as they flow across multiple different chemistries. So a lot more work and probably a lot more development time to get a product to market if you're going to move away from the traditional line and even multiplex line dispensing and going to the array pattern.
I think just because you can doesn't mean you should. Is that?
Yeah. Exactly. It's kind of like exactly what I tell everybody as well. I mean, it sounds really exciting and you think that you're going to come out with something novel and very unique, but there's a reason you don't see a lot of that on the marketplace just yet. You have to have the volume and the quantity to really justify the capital equipment needed. And it's a different level of capital equipment investment, which could be maybe 2x your traditional. So you really have to think it through.
Yeah. I think it's so important. Design for manufacture. I think there's a lot of time spent on the front end on something exciting and new and, for lack of a better way to put it, cool. And then when people have to, the rubber hits the road and you have to say, okay, now what is this really going to cost on the end? That's where, and a lot of times it may not make sense to be that fancy.
It's important for us as a company to help steer the customer to fully understand what it is they're asking for, because the last thing we want to do as a business is provide equipment to somebody and then have them not be successful. I mean, they're investing with us to be their partner, and so we want to treat them as such.
Yeah, I agree very much. Well said. So speaking of wanting your client to be successful, where does variability tend to show up during the handoff from a benchtop to a pilot lot? And what is the earliest signal that will affect throughput or yield?
It can be difficult to answer this question, but I'll kind of tell you some of the challenges that we see from the equipment side of things. And that is when you're developing on the benchtop, the traditional and easiest way to do your R&D work is to utilize what we call our contact frontline dispensing tip. And it allows you to use smaller volumes of reagent, do a lot of benchtop testing. But when you do utilize the contact dispensing technology, it's basically a drag needle, right, being rested on top of the nitrocellulose. You do end up potentially scoring the surface of the nitrocellulose. And then naturally, if you can envision this, is nitrocellulose is a cellular paper structure. And so the surface is inconsistent. And so the tip will follow the cellular nature or structure of the surface, and it might make the tip move a little bit more. So you might not get a line that's as crisp as you would using a non-contact dispensing technologies. And so what I see is a lot of people just want to jump from one to the other or vice versa without really having completed all their work in evaluating which process actually develops the highest sensitivity test for them, right? And you can't always say that contact printing is going to be worse than the non-contact biojet dispensing. We do see better performance traditionally, but sometimes it's not measurable enough to make that leap. And if you don't need to move from a contact dispensing technology to a non-contact dispensing technology, it's best to go with what you were traditionally developing with and what was already working. The second part that I wanted to really answer is when you're doing benchtop development work or small scale manufacturing, right, you have a user that's literally looking at and monitoring. Sometimes, or most of the time, they have a Sharpie pen and they're trying to visually inspect the quality of the line dispense that's occurring on the striping. And if you get an air bubble in the line or for some reason there's some inaccurate dispensing that occurs, right, you need to mark that section out. You need to somehow discard of it because it's non-usable and won't work downstream if you were to include it as part of one of your strips that you're putting into a cassette. So the biggest benefit of moving from benchtop to inline is the fact that we integrate inline QC or a vision system to monitor and bad part mark anything that falls out your parameters. One, the vision system can actually measure the thickness of the line. It can look for breaks, bulges, satellites. It can also measure for position of the line from the bottom edge of the membrane, because positioning and spacing from line to line can be very critical in the performance of your test as well. And so all that's done automatically with the system, and so your yield goes up tremendously when you're using a vision-based system to do your monitoring rather than an individual using a bad part marking Sharpie to note that, right? And I think those are the two big components we really drive home on the equipment side of things so that people are aware. That would be my recommendation for people trying to pay attention to those items.
Thank you for that. That's something definitely to think about. I appreciate it. So what does a great first 90 days on new equipment look like? Give us a checklist that helps teams stabilize quickly.
Well, it really depends on if it's a batch system, right, benchtop, or if it's a big inline system. So I'll start with the inline one because the answer to that question is you're probably not getting much done in 90 days, right? I mean, we can get the equipment installed. We'll do installation and training on site. We usually spend a week or so at the customer site to do that. And then most people will also have us do what we call an IQ, OQ on the instrument, installation qualification, operator qualification. And then we sign off saying that the equipment is meeting all manufacturer requirements, and we hand them a binder so that they can move on to their own PQ work. But that level of work is, the success is getting the equipment ordered, installed, and qualified in the first 90 days. But usually it takes, many more months after that before you can validate the instrument and actually run actual product on it. Now, going back to R&D level, right? These are small benchtop dispensing platforms. They're much easier to set up. They're much easier to get up and running. And usually you don't need to qualify them to any great extent unless you move them from R&D to, medium scale manufacturing. And so the first 90 days would be installation, training, and us mainly making sure that you understand, all the settings and parameters so that you can operate the equipment on your own and be as successful as possible. But for most of the manufacturers, right, unless they're an experienced user, they're just getting their feet wet and they need to spend some time with the equipment. So you're going to make mistakes and you just got to try it and do it, right? I mean, there's nothing like on the job training, so they say.
Absolutely. I'm going to go back a little bit just because now I'm curious. You made a statement, as long as you're just talking about R&D and you're not wanting to use the equipment for medium scale manufacturing. And as we discussed earlier, there are probably more diagnostics at scale that could be run on this benchtop than maybe in line. So Tell me about what has to happen for you to move from R&D to manufacturing for the benchtop.
Yeah. So similar to reel, actually. So at some point, you need to put them into a manufacturing environment. So you would no longer utilize the same piece of equipment for R&D work. Traditionally, you either buy a second set of equipment that's dedicated to manufacturing only and leaving your R&D system for the R&D group to continue to manufacture additional future tests, but you would transfer everything over to your next set of benchtop equipment. And when you do that, you then need to qualify that equipment. So you would need an IQ, OQ. You would need to get the equipment validated and set up in a manufacturing lab environment. And that would be the big difference between the two. The equipment doesn't get any more expensive. The equipment is identical. It's just you have to make sure you go through the proper quality channels, and establish your processes before you make that jump.
Makes a lot of sense. Based on user feedback, what do customers end up liking that they did not expect at the time of the purchase?
There's a few things. The beauty of BioDot's dispensing platforms, and they probably don't realize this, but we put a lot of forward thought into making the system versatile enough to grow with you. Okay. So you don't want to buy a piece of equipment or make this level of investment without the system being flexible. And so what I mean by that is a lot of people start off, they're just getting started in a lateral flow market and developing, they've got an ELISA assay that they're just going to seamlessly transfer over to lateral flow, right? They need one test, one control line, and they need to spray their conjugate. So they need one air jet for spraying their conjugate. And what they don't know is in the future, they're probably going to do additional test lines or more complex tests, right? And so the beauty of all our systems is that we can allow anywhere from one dispensing channel all the way up to 7 on a given platform. And you don't need to do anything other than plug in a new dispensing channel onto the system. So the system will grow with you and can adapt. And that's really only speaking to the benchtop system. The reel-to-reel dispensing platforms and our inline auto lamination system, something that's extremely unique to that of the competition is all our modules on the reel-to-reel systems are able to be added in or removed at any time within the system. So let's say you don't have budget and you want to just stripe one test, one control, spray your conjugate, and you want a single dry tower to save money because you don't want to run 2 or 3 drying stations. You can start off with that, and all you have to do is run your equipment at a much slower rate of speed, 10 millimeters, 20 millimeters, maybe 30 millimeters a second. And then if you want to continue to scale, you can just plug in another drawing tower, and then you can bump up your run speed on the system to double the speed to produce twice as much product, and you now have the drawing capacity to handle that. Or another example would be you didn't know that you were going to need to dip your material or do a blocking step, and you didn't buy a dip tank. Well, you can simply unlock a couple of the modules, push the system apart, kind of split it down the middle, if you will, plug your dip tank in line, and now you're running with a dip tank. So the system's highly flexible and can be modified to really adapt over years and years of use. And so it's nice when a customer buys a piece of equipment from us, gives us a call a year later and says, hey, we need to do something totally different. Is this possible? And we go, oh yeah, it's already been built in and designed to your system. And they go, no way. How do we do that? And we go, well, we just simply, order the extra components, let's get them installed, and you'll be off and running. And so it's a nice surprise, and customers really appreciate that in the long run.
That's fantastic. For someone who would be brand new and be fearful of the investment going in, what you're basically telling me is that they can't box themselves in.
Yeah.
They don't have to fear that. That would certainly be a pleasant surprise.
Yeah, that's my favorite one, for sure.
That's fantastic. So let's switch gears just a little bit.
Okay.
You've hired and coached sales teams for technical products. What backgrounds ramp the fastest when selling into diagnostics manufacturing?
A lot of people would say maybe you need a scientific background, but I actually believe the opposite. So, it's nice to have a scientific background and maybe some experience on the workflow that you're trying to address. But in the manufacturing and automation environment, that's less critical a lot of times to the customer. They really want you to understand the hardware and the automation side of the workflow, and how your equipment can address their issues and concerns. So traditionally, when I'm coaching, hiring, or expanding a sales team, I'm looking for people who have capital equipment automation backgrounds. And then if they have some science behind it, then that's almost a slam dunk. That's like a really nice, addition. I kind of put the fact that, they need to have some mechanical aptitude and some automation experience to really make sure that they're able to service the customer properly.
What is your advice for early career professionals who want to work at that biology hardware boundary?
I think you have to be willing to take an entry level job, and you can do that in many ways, right? You can become a bench scientist and work on developing some of these assays, or you could actually start in a junior role where you're assisting salespeople in locating, finding, and hunting for new potential customers. And during that process, you can slowly migrate out to being in front of customers and building up, your repertoire of sales skills. There's nothing better than experience. Unfortunately, there's no silver bullet. You just have to do it. You have to put in the time, the energy, the effort. I have a couple big statements that I use all the time when I'm interviewing people or giving recommendations, and that is, I need to know that you care and you actually want to learn, right? So if you don't care, I don't think you're going to be successful. So pick an industry that you really like, that you can invest in, you feel is maybe benefiting the world in a larger way than just you obtaining a paycheck, right? And you're probably going to put in the work and the energy and the time needed to be successful. It'll take years of work before you get to maybe, the level of a sales manager, in an outside role handling a large territory or region.
Yeah, you have to have purpose, right? The why.
Purpose.
Why? Yeah. Why am I doing this? I'm sure, Rob, you're also going to highly recommend that they attend DCN's basic lateral flow training.
Oh, absolutely. As I mentioned, there's nothing better than on-the-job training, and you guys have a nice course down there in Carlsbad that you put everybody through. You got, I think it's 3 days of hands-on training and experience that they get working with Biodot equipment. Thank you very much, DCN. Yeah, I highly recommend attending that course if you're just getting started in lateral flow. I've been attending the ALFC for as long as I think I've been at Biodot, and we've been a major supporter and sponsor of it. We are there every single year. It is by far the preeminent workshop for lateral flow in the world. You may or may not know we do some workshops ourselves in Europe. Biodot does a little bit different style than the advanced lateral flow course, a little more hands-on. But we don't do that here in the States because you guys do it so well. Why don't we just come along for the ride and support you guys? But yeah, it's a great place to learn and get to know everybody. My biggest pitch for anybody who's in lateral flow and wants to learn more about lateral flow, it's not just going for the courses and the presentations that are given, but it's actually meeting all the varying community members that make up the lateral flow industry. They're all at your event, whether it's the nitrocellulose, right, manufacturers, the equipment manufacturers, but you need to know everybody in the industry to be successful, and you get to connect with everybody all in one foul swoop.
Absolutely. I was really happy to see that you and Biodot will be at ALFC in October. What will Biodot be emphasizing at the event, and who should come talk with you on site?
Yeah, so naturally we're emphasizing our lateral flow, striping, spraying, and cutting technologies, as well as lamination. So if you're in need of benchtop striping or reel-to-reel inline high volume manufacturing, then we'll be there with information on all those items. It's mainly, we are here to answer questions and let people know about our product.
That's great. Is there something they should prepare or think about to make the conversation with you more productive?
If they have an idea of their test and what they're trying to accomplish, right, whether it's striping lines or doing array patterning onto nitrocellulose, number of unique chemistries we need to work with, right? So number of test and control lines or dots, how they want to potentially spray their conjugate onto the glass fiber. If you know those basic things, then we'll easily be able to direct you on the proper system setup and configuration. But it's mainly number of test and control lines is really the starting point of the conversation.
Okay, that's great. Thanks for sharing that with our listeners. Rob, thanks so much for joining us.
Yeah, no problem at all. I enjoyed it so much. Glad we got to cover all these topics, and hope you guys have me back again.
Thank you so much. Absolutely. And for our listeners, we'd love to see you at the Advanced Lateral Flow Conference 2025 in La Jolla this October. You can use code ALFC25 to save 25% off any registration type. Visit alfc2025.com for details. And if you plan to be there, be sure to stop by Biodot's booth and say hello to Rob.
Yes, come on by.
Thanks again for joining us on another episode of Expert Insights. We'll see you next time.